Thank you. The 10th is a soluble form of adenylate cyclase. And that 10th soluble form of adenylate cyclase is phylogenetically the oldest of all the adenylate cyclases. This is probably the original adenylate cyclase. And interestingly, it is found in cyanobacteria and is related apparently to carbon dioxide sensing. So in the cyanobacteria, which remember are photosynthetic bacteria, right? They don't use chlorophyll. They're not green. They're more cyano-colored, right? And the adenylate cyclase's function was to transmit to the molecular machinery of the bacteria information about CO2, as far as we know. Again, this slide this just showed you a breakdown of all these different families, and we'll come back to that in a minute. If you look in general at the 9 adenylate cyclases, they have 6 transmembrane alpha helices, so it's going to go through the plasma membrane 6 times. And then they're going to have 2 cytoplasmic lobes, which make up the catalytic site. Depending on the group of alpha helices, they're either going to be C1 or C2. And the catalysis occurs in a crevice that's formed by the 2 of them, okay? And let's look at that. So this is a little misleading, but helps you get a sense of alpha helices. So this is 3 alpha helices pairs that are going through on each side. This is the C1, this is the C2. This is going to have to come in contact with this in order for them to make the active site. So this sort of extended thing is nonsense. It's just to show you sort of to see what's involved here. I'll show you a better picture in a second. This picture is nice because the green is when it's not in contact, when it hasn't been activated by the G protein, and the purple is what happens when it is activated by the G protein. So it just shows you how these alpha helices actually move in response to the binding of to the alpha stimulatory subunit. Here, this is a better 3-dimensional rendering of what this thing actually looks like. So 3 of the alpha helices pairs are grouped together almost in a box, a cube, a rectangle, a 3-dimensional rectangle. And here you have the same thing here. And then these are their 1 and 2 chains, and the crevice in here is where the active site's going to be. So this picture is a pretty good picture of what's going on. We're going to look some more at that in a little bit. All of the membranous adenylate cyclases are activated by the stimulation of G protein coupled receptors and the liberation of the G protein, GTP, G protein alpha stimulatory subunit. You remember that there were several alpha subunits, right? And some of them were inhibitory. We're going to see them. Some of them were often photoregulation. Some of them were in olfaction. But the cyclic nucleotide, the adenylate cyclases, are activated by the G-alpha stimulatory. And this is a molecule, forskolin, which you're going to see the term forskolin, and then it's activated by forskolin. If you read the literature at all, it's not a physiologic compound. It's a pharmacologic tool. But I thought you should just know about it. And this, again, this is a very good summary. In comes the hormone, binds to the G protein coupled receptor, activates the alpha stimulatory unit, goes and interacts with the adenylate cyclase, changes its conformation, and you see how they've drawn it as 2 lobes, changes the conformation, and then that takes a typical ATP with its 3 phosphate groups, all right, and makes cyclic AMP. In more detail, so here you have epinephrine binding to the G protein, the beta adrenergic receptor, the G protein coupled receptor. GDP gets thrown out, GTP now bound to the alpha subunit, interacts with the adenylate cyclase, makes cyclic AMP. And now in this slide, we see the downstream effect of the cyclic AMP, which is to activate, primarily, its primary function is to activate protein kinase A. Protein kinase means it's going to phosphorylate another protein. That's what a kinase is. So it's going to phosphorylate a whole world of proteins, right? Just a whole world of proteins, depending on lots of things that are specific to unique states and unique situations and unique original primary transmitters and unique positions of all of this within the plasma membrane. And just keep building up now through these cartoons more and more complexity, because this was what we just looked at. So this, the left half, from adenylate cyclase to the left, is what we just looked at here on this slide. So from the right of the adenylate cyclase is the inhibitory side. So just like you have the stimulatory side, you also have an inhibitory side acting through the G-alpha inhibitory subunit. And they then integrate, okay, to regulate the enzymatic function of adenylate cyclase and how much cyclic AMP is present in the cell, how much cyclic AMP is synthesized. Because the other side of regulating this, which we won't come till the end of this talk, is how fast you degrade the cyclic AMP. And you may think that this looks complicated. Let me tell you something. The phosphodiesterases are much more complicated. The ones that are responsible to degrade the cyclic AMP are more diverse and more regulated than the synthesis. This is a complex system. But it is, as I say down here, this is wonderful. If you've got diverse systems coming in and you want to deal with sunlight and your degree of arousal and whether you're hungry and whether you, and all those things, and you want to integrate all these very diverse things. So, you know, just letting them all feed into the level of cyclic AMP in the cell is not a bad way of doing things. It's a good integration system of diverse signals. That's what it is. Again, in the adenylate cyclase, we said there are lots of isozymes. There's different differential regulation of various isozymes. They are all tightly controlled. They have subcellular localization. The adenylate cyclases, think back to the cytoskeletal system that underlies the plasma membrane, the microfilaments on the actin system. So you could move these adenylate cyclases. You can assemble them into rafts. All the things that we've previously talked about apply to the adenylate cyclase, all these things that we've talked about. But you can also stimulate it. So the group 1 of the adenylate cyclases are, there are several of those, but all of the ones in group 1 are stimulated by the alpha subunit, but they're also stimulated by calcium calmodulin, which we're not going to talk about much today, but we're going to have a separate lecture on calcium and calcium calmodulin and calcium-dependent kinases and everything else. And they are found primarily in the cortex, the cerebral cortex, cerebellum, and hippocampus. Group 4 also does that, as well as the olfactory system. They are activated by catecholamines. So your whole dopaminergic system is going to be using this kind of thing. They're activated by hormones. And they're also activated by a totally different second messenger system, the protein kinase C, which is a separate lecture that we will talk about. So that's going to feed back. Group 1 is what we're talking about right now. So just stay with this for a minute. This really just, here's your alpha stimulatory, here's your adenylate cyclase. Here comes calcium in via calcium calmodulin. That is also can activate it. You can also use the beta gamma dimer of the G protein to inhibit it. And strangely, although calcium calmodulin activates it, it also activates calcium calmodulin kinase, which then can feedback and inhibit. So you've got feedback systems, which, of course, which you need in order to be able to terminate signals. You also have other groups, the group 2, and this is a level of detail that's way too much. This is more illustration and giving you a sense of the breadth and complexity that you will encounter, but there's no point in trying to remember any of this stuff until you're ready, until you encounter it, because you're talking about the D2 receptors when we talk about dopamine stimulation sometime in January. Then you will be using one of these mechanisms. That's when you'll get specific. We're not trying to get specific, we're just trying to show you the palette that this system has available to it, which is incredibly diverse. Don't try to remember this stuff now. Group 2 is involved in olfaction, but it's elsewhere throughout the brain. And group 2 has no sensitivity to calcium. So you might not want your system. You might want to use a cyclic AMP system, and you might not want it sensitive to calcium. Fine. Okay, good. So you have one of those. And notice that group 2 is activated by the beta gamma subunit, and group 1 was inhibited by the same subunit. But that doesn't mean that those two things will happen simultaneously, does it? Because the group 1 adenylate cyclases may be in the proximal dendrite, and the group 2 may be in the distal dendrite. So you have spatial segregation. Okay, so all of this could be going on at the same time, and the same release beta gamma subunit may be tending to activate part and non-activating part, and that's where the phosphodiesterases are going to get really interesting. Group 3 of the adenylate cyclases, you can read them. This is the type that you find in the striatum. This is what we'll be talking about here. And then group 4, as I already mentioned, have the same anatomic distribution of the group 1, although that doesn't mean at the subcellular level they're going to be the same. They may not. Okay, and these are inhibited by calcineurin, not calmodulin, but calcineurin. Okay, so this just sort of summarizes. You have different groups. They are all stimulated by different things. Look at how simple group 2 is. You just stimulate the system. Fine, I get it. Ordinary stimulation, and protein kinase A inhibits the adenylate receptor. But that was the target of the cyclic AMP. So this is just a simple example of that, direct and immediate negative feedback, period. So as I say, but this one, these two, very complicated. Okay, very complicated. So you make the cyclic AMP, and you increase the cyclic AMP concentration in the cytoplasm. Then what? All right, well, then most of the time what you're going to do is you're either going to activate protein kinase A, or you're going to interact directly with these cyclic nucleotide gated channels. All right. The easiest one to think about is protein kinase A. It encounters cyclic AMP, and in a mechanism that we'll look at, the complete protein kinase A is complexed, usually to the membrane, with A kinase anchoring proteins. They hold the cyclic AMP, they hold the protein kinase A sequestered next to the membrane. And then you bind the cyclic AMP to the regulatory subunit, which then dissociates the complex, lets the protein kinase A go because the anchoring proteins were stimulated by the cyclic AMP, and it releases it to become active. And then once loose, the protein kinase A, as a kinase, it phosphorylates anything that it wants, okay? AMPA receptors, ion channels, cytoskeletal proteins. It can feed back on other adenylate cyclases. You find yourself with just this remarkably complex and beautifully integrating system. Complex for us to learn, beautiful for us to see how exquisitely tunable the system is. That's really what we're getting down to, how tunable the neuronal signaling mechanisms are for integrating information. Leaving protein kinase A, you have these are ion channels that are gated directly by cyclic nucleotides. Wait until you see cyclic GMP, okay? And you have these hyperpolarization activated cyclic nucleotide gated, okay? Olfaction, pacemaker activities. That's why you'll see this pacemaker activity. You'll see the one type in the suprachiasmatic nucleus, which is the circadian clock. So this is ideal for pacemaker activity to do this sort of thing. You let the channel open. You let it run for 12 hours if you want. Meanwhile, there's a molecular clock running, maybe in the nucleus, maybe running somewhere else. That clock goes off where you're exposed to light, or the melatonin is degraded, and the phosphodiesterase comes in, destroys all the cyclic AMP, and the clock is reset, just like that. Lovely feedback mechanisms. Again, I mentioned that's the broad beta adrenergic stimulation of the frontal lobes that occur for decision making, for thinking, for making up your mind under situations of more or less stress. You change that decision making process by the degree of beta adrenergic stimulation that your frontal lobes are experiencing. So when we talk about decision making in the prefrontal cortex, we mentioned that it's highly dependent on what else is going on the state of agitation of the organism. This cartoon I ran across this morning, I liked it a lot. This is the how cyclic AMP controls protein kinase A. Very simple cartoon, but I like that for its simplicity. Here is, we're going to get more complicated with a minute, but here you have a regulatory subunit holding on to the catalytic subunit. 4 cyclic AMP molecules bind to the regulatory unit. Then the regulatory unit releases the catalytic subunit, which then does its thing. And because the catalytic units are quite small, they also can be taken up into the nucleus, and a lot of the influence of these is going to be at the nuclear level. We'll look at that in a minute. So this gets more complicated, right? But it also summarizes what you already have seen, cyclic, the neurotransmitter, the external signal, the G protein, the G protein receptor, the G protein, the synthesis of cyclic AMP by adenylate cyclase. Now that comes in contact. Look at over here on the right. This is a great cartoon. Here's the whole complex. Here's the anchoring protein, here's the regulatory peptide, and here's the catalytic subunit. You bind cyclic AMP. When that goes up, okay, it doesn't bind to any of these parts. It binds to a regulatory subunit in association with the anchoring protein, and it causes the anchoring protein to let go. And once they're let go, the cyclic AMP interacts then with the regulatory subunit, which then causes it to release the catalytic subunit, which phosphorylates its cytoplasmic targets or goes into the nucleus. This is the whole picture here. Now you're seeing the whole thing. And remember, here's your plasma. You lose sight of the plasma membrane. It's just like, you lose sight of the nose because it's there all the time. But every time you draw these pictures, it's sitting in a bilayer, okay? Which was the point of our first lecture. More detail, same information, more detail, different system, okay? And here's an odorant molecule, an olfaction, much more direct than with a neurotransmitter, but still it's a chemical signal of a G protein coupled receptor. And the same thing happens, and eventually you increase the cyclic AMP, and it stimulates kinases in the cytoplasm and in the nucleus. And it might also, the cyclic AMP might also directly activate a nucleotide gated channel, which then changes the resting membrane potential directly. All of this being done through the second messenger system. When you look then at what the cyclic AMP signaling does, just look at this, and I'm going to ask you a question. What do all these things have in common? Synaptic plasticity, learning, memory, pain, dopamine stimulation, enhancement of glutamate agonists, antagonism of endorphins, olfaction. Well, maybe that's not fair. It's going to be even more obvious on a later slide. So let me just tell you, these are all long-term. These are not quick. These are not a fraction of a millisecond, okay? These are setting things, learning, okay? Memory, the dopaminergic activation of the striatum. These are not momentary things. This is, you're going to walk across the room, You're going to be awake and active. You're not going to be asleep, okay? Pain regulation is going to be, this group's going to be really important. So, and that shows up here in that you have the long-term regulation of the adenylate cyclases through gene mechanisms, provides a mechanism for downregulation of drug receptors. Okay, so this is a good example of how tachyphylaxis can occur, downregulation. And if you continuously use long-term receptor agonists, so you keep it up, you just keep stimulating them, you're going to eventually lead to a desensitization of the system by a feedback mechanism into the nucleus. All right, and the same is true if you long-term if you long-term antagonize the system, you're going to make it more sensitive. And then the real problem occurs, right, when you change the state. So you've been stimulating the system with Cinnamon, right? It's down-regulated itself, and you remove the cinnamate, you give them a drug holiday. They're rigid. You've down-regulated the system because of the chronic stimulation. Or turn it the other way around, right? You have been blocking the system with Haldol, right? And suddenly release the system by taking the Haldol away. And they're super sensitive. So any dopamine that stimulates, and they get all kinds of movement disorder, tardive dyskinesias and things like that. So this is the mechanism by which all these things occur. And the same thing occurs with the opiate systems. It's the same concept. And that is, the more you use the opiates, the more you downregulate the system. The more you downregulate the system, the more you need the opiate. Okay? And where is this, do you know where what the lethality of the opiates is? You know where the deaths are occurring? So the heroin deaths are nearly all occurring in people who have recently kicked the habit. Okay? So they've gotten used to a certain dose over a year, 2 years, right? Then they say, enough. I don't want to do this anymore. They get clean. Okay? Then they're out at a party and someone says, yeah, we haven't seen you in a while. Come on in the back. We'll do this stuff. And he takes the dose that he used when he finished, and he's dead. Okay? Because that dose was to a desensitized system. And the system is now back to normal, is no longer desensitized, and he takes that same dose. But the system is not capable of dealing with that dose. And that's where the people die. It's very consistent. That's when the heroin, I'm talking about, Percocet or the other thing, but that's where most of the heroin deaths are occurring. Just that simple story. Okay, all of that is cyclic AMP. Let's turn our attention to cyclic GMP. We have plenty of time, probably more than halfway through already, because we can build on what we learned, although there are some important differences here. First of all, just like with cyclic AMP, you make cyclic GMP from GTP. The cyclic GMP also regulates lots of proteins, kinases, ion channels, and phosphodiesterases. Time for the phosphodiesterases. You have two major classes of cyclic GMP activated proteins. All right. The first is of the guanylate cyclase. You have a membrane-bound group that will look sort of like adenylate cyclase. They are pretty regularly activated by the natriuretic peptides. So when you see BMP, brain natriuretic peptide, that's actually not brain. It was first isolated in brain, but the NP of brain is CNP, not BMP, strangely. That's the way it goes. History is sometimes cruel to naming. And then there's a second group, which is really, it isn't that there's no analogy, there is. The soluble adenylate cyclase is reflected in the soluble cyclic guanylate cyclase. But here's the difference. The adenylate cyclase in humans and mammalians is sort of overwhelmed by the 9 membranous forms. But at the guanylate cyclase, this is a really important enzyme. So this little soluble form is not just a minor afterthought, okay? This is a big deal when you're telling about the guanylate cyclase. I also just added this morning, and that is I want to point out that both soluble forms, both the adenylate cyclase and the guanylate cyclase, both soluble forms are activated by gas. So they are gas signaling. The adenylate cyclase was bicarb. Okay, the guanylate cyclase is nitrous oxide, nitric oxide. Nitric oxide, as you know, is not trivial. Okay, this is a major part of regulating everything, everywhere. Okay, because it's a major vasodilator, but it's also a major signal, and so we're going to look at it. Now, the other thing that's really different about guanylate cyclase, really different, is, well, look at this slide. Here's your guanylate cyclase. What's different about it compared to adenylate cyclase? Take a look. Tell me what's the difference. Right, there's no G protein. Okay, so this is directly stimulated by the ligand. There is no receptor, G protein coupled receptor. That and the G protein are not here. Okay, in this case, the ligand interacts directly with the guanylate cyclase. Okay, so otherwise it looks sort of similar. Now, what's the consequences of that? You don't get as much control. The adenylate cyclase is an amazing system for integrating because you've got all these myriads of receptors which you can make independent of the G protein. But the guanylate cyclase is not that way. And so you're going to have a much more limited, I use the word palette every now and then, much more limited palette of the types of things that you can do with guanylate cyclase. When you look at the, this next slide is nice, but there's plenty of diverse actions. So here's your plasma membrane. Okay. Here's the external port of this receptor. Notice that it's a dimer. Okay. The dimer has three components to it. Ligand binding, a regulatory and ATP sensitive regulatory, and the catalytic subunit. And they all lead to cyclic GMP. This we're not talking about right now, but we will in a little bit. This is your nitric oxide sensitive soluble, which is analogous but a little bit different. Instead of a receptor, you have the NO sensor, which is basically a heme group. We'll come back to it. The receptor domain is for extracellular signaling. We've been through this. The receptor binding leads to oligomerization, which that combined with phosphorylation, ATP, enhances the generation of cyclic GMP. B type does occur in neuronal tissue, but so does the C type, okay, natriuretic peptide is the primary one in brain. D and G are in olfaction, E and F are often photoreception. And I can't decide if I like this slide or not. Here you have a G protein coupled receptor. You're making cyclic GMP. You then activate protein kinase G. You knew that was coming, which then phosphorylates whatever it wants in the cytoplasm or in the nucleus. And then over here on the right is the nitric oxide stimulation to create the cyclic GMP that way. And you can see that they have different effects. And we'll look at this. This is, I just put this slide up here because this is all of the NPs, if you will, the natriuretic peptides, have a common structure of a 17 amino acid peptide ring. So I don't know what the significance of that is. I haven't studied what its configuration is or anything like that, but I thought I'd just include that the diversity of the peptides are in these side chains, but they all have this rather seriously conserved 17 amino acid ring. And then I think we've used this slide before. This is just an example of phototransduction. Instead of using a neurotransmitter for the protein, for the G protein coupled receptor, light can trigger a conformation change, which leads to the same cascade. And here, this slide, what's good about this slide is that it shows you that this also integrates with the guanylate cyclase system. So now the cyclic AMP and the cyclic GMP are all interacting with one another. Turning our attention to the soluble guanylate cyclase, we said it's similar to the membranous, but I love it. It lacks a membrane domain, very cute. The enzyme activity still resides down in the same subunit. It is activated by nitric oxide, which binds to a heme group in the amino terminus of the alpha and beta subunits. And of course, the nitric oxide is synthesized by nitric oxide synthasase, which you have constitutive and you have, that is, you have constantly ongoing and then regulated NOS. And all of the NOSes convert L-arginine to nitric oxide and citrulline. And type 1 is known as neuronal because there's a lot of it there. And this is a good cartoon. So here's the, over on the right, this is the membrane-bound form that we've been talking about. But on here in the center is the soluble form. And you can see this heme binding to the regulatory subunits, which then control the central domain. And finally, the catalytic subunit. And we saw this slide before, and now this is just to put it all in perspective of the two of them together. The downstream is primarily through protein kinase G and G protein, a GMP-gated ion channel. So very similar to adenylate cyclase, very similar in that way, which would have been protein kinase A and the cyclic AMP-gated ion channel. So very similar in that way, and both of them feed back on phosphodiesterases. And the protein kinase G has just as diverse a group of proteins that it phosphorylates and therefore regulates, up-regulate, down-regulates, in between, okay, including all these things, which we'll be looking at, phosphatases and calcium signaling. So when we, next week or the week after, when we start to look at phosphatases and calcium signaling, you have to remember that these are regulated by the cyclic nucleotide pathways and in turn regulate the cyclic nucleotide pathways, all of this being orchestrated through the G proteins, all of it, okay? And I like this slide. Again, I just use a lot of cartoons. Here's the protein kinase G as the focus of this slide. And you can see that it might inhibit the endoplasmic reticulum, or it might inhibit calcium entry, or it might inhibit the further guanylate cyclase, so it stops making it. So this little loop here would make a negative feedback mechanism. But it also can stimulate PDE5, which will then degrade the cyclic GMP. It might also directly phosphorylate NOS, which might make it more sensitive to the presence of nitric oxide. All of those are possible. Now, I want to just take a moment. You're going to hear about, so I think this is a great pair of slides, and I think of images. And this is, you're going to hear this, and this to me has always been a mystery. And it's no longer a mystery. It's so simple. So the topic is the leucine zipper and the zinc finger. So what the hell are a leucine zipper and a zinc finger? And what's the point? Okay, so it's so easy, okay? Think about an enzyme. Okay, and think about adenylate cyclase. If you go back to that slide that I didn't like, where you had the adenylate cyclase extended through the plasma membrane, and you had these two catalytic subunits that were far away from each other, and had to be brought next to one another. This is not a trivial move. Okay, this is not somebody moving 1/10 of an angstrom over to interact a little bit more readily with a magnesium ion that's sitting there in the active site, right? This is, you got to move some whole peptide chains around to make this thing happen. So you're going to need a lot of energy. Okay, so the small configuration change, a little energy. Big configuration change, a lot of energy. So how do you use, you don't want to have to, you want to be able to amplify your signal, okay? You want to amplify the energetics of your signal. So on binding of a small molecule of cyclic GMP, which isn't a whole lot of energy when it binds, right? It's going to induce some very small configuration change. You need to amplify the energy involved in this. That's what the system does. So you've got the zipper, okay? And when you bind to it, the zipper opens. That's a lot, okay? And that's not enough, okay? The zipper is controlling a zinc finger. And a zinc finger is like a big iron clasp. And so when you run the zipper, this thing moves, okay? This is a lot of force. So if it happens to be holding two peptide chains and the zipper runs down and it closes, it brings two whole catalytic subunits into approximation to one another. So the leucine zipper and the zinc finger, which are really well shown on this picture, their purpose is to amplify the energetics of a small signal to activate a process which requires a fairly large activation energy in order for it to occur. Okay, so the leucine zipper and the zinc finger amplify the energy of the signal. They don't necessarily mean that more is going to happen. That's a different process of amplification. They amplify the energetics of the second messenger binding, okay, via this mechanism, which I think is very cool. And that's just so that really demystified that for me. That's pretty clear. This is where I think when you look at cyclic GMP, this is where I meant to ask, what do they all have in common? These are all long-term events. There's nothing in here short, right? Everything is long-term. Okay, as we come to the end of this, then we want to, so we've been looking at adenylate cyclase, we've been looking at guanylate cyclase, we're making cyclic AMP, we're making cyclic GMP, now we have to get rid of them. So like the synthesis, these are going to be highly regulated. That's an understatement. So we thought that we had a lot when we had 9 different families of adenylate cyclase. There are over 50 isozymes of phosphodiesterases. Over 50, okay? That's an astronomical number, okay? And there's a reason for it. They've evolved for a reason. So this is an area of extreme interest right now in pharmacology because, of course, in pharmacology, you want finer and finer tools. The more you can regulate one process and not spill over into unwanted, you know, what do they say, unintended consequences, the better you like it. And so the more diverse the family, the more likely you are to be able to interact in a very subtle, sophisticated way. And whereas and even have 9 adenylate cyclases, that's a lot, okay? But on top of those 9 adenylate, 10, counting the soluble form, you have 50 phosphodiesterases. 21 different genes are involved, and you have alternative splicing. This is an extreme customization. They have 3 functional domains. There is the N-terminal regulatory domain. That's where all the variability is. The catalytic subunit is the same everywhere. All you're doing is breaking a phosphodiester bond. And that's not hard to do because that's a queer configuration, right? A phosphodiester bond. We don't encounter that very often in neurotransmitters and peptides. And that's a very peculiar bond, that phosphodiester bond that we saw. There's nothing that looks quite like that anywhere. So you don't need a lot of different catalytic subunits to break this. What you need is the control of that catalytic subunit. Okay, so here's a good picture making cyclic A. Here's the synthesis of cyclic AMP. Here's the degradation, very simple. And the point is, though, and here's the same thing for cyclic G, but the point is these are going to get integrated into very complex systems very quickly. This is just a taste of the variability of the regulatory subunits and how little the catalytic subunit changes. I mean, that's really all. It's just an illustration. You have calcium calmodulin stimulated, you have them, there are all sorts of things. These are active, this is a major group. They are activated by, in the physiologic range of calcium. You find this type of phosphodiesterase up in cortex and hippocampus, which, as you remember, were group 1 and group 4 of adenylate cyclases. And here the PDE1B is in the dopamine stimulated tissues such as striatum. You have G protein activated phosphodiesterases, okay? So in other words, this provides a feedback mechanism, and you see that in retinal phototransduction. You want to turn the signal up fast. You have phosphorylation regulated. So these are feeding back on themselves. They're phosphorylating themselves. You get on. And that's what I said. This is where people are, the pharmacologists really are hoping for highly selective pharmacology here. Unlike caffeine, which is a very broad in a group. Sildenafil is PDE5, okay? PDE4 is being explored. PDE, these are the phosphodiesterases. Be clear that we're not synthesizing. We're looking at controlling, pharmacologically interacting with the degradation of the cyclic nucleotide signal, okay? So we're working at the turning the system off. Okay, being explored actively for memory and cognitive disorders using the heart. Dipyridamole and platelet interactions is a phosphodiesterase inhibitor. This diagram is just, it integrates the different types of PDE, phosphodiesterases, into the system. Again, sort of building it up, adenylate cyclase, guanylate cyclase, nitric oxide synthetase, all that in coming together. And heading toward this idea that I've been talking about from the beginning today, and that is that they now, with the phosphodiesterases, you can create, the cell is capable of shaping gradients of the concentration of cyclic AMP. So I might have a nidus. Think about for a minute. Let's just, I don't know if this is true, but just as a simple thought, okay? Let's think about an axon, a myelinated axon. And you have a node of RON-VA and you have a node of RON-VA. And they're going to be the active zones. And in between, you don't want active, right? So put a cyclic nucleotide here. Put a cyclic nucleotide, so that is, put an adenylate cyclase. I'm making this up, okay? An adenylate cyclase at the next node of RON-VA. And not far away from the node, okay, assemble, right? Use your cytoskeletal system to assemble a collar of phosphodiesterase. That will restrict the cyclic AMP to the region of the node of RON-VA, because as soon as it might tend to diffuse down the axon into the myelinated segment, and you don't need, what for? You don't want it to do anything in there. You want that area to be as energetically favorable as possible, meaning as quiet as possible. So you don't do it. You put a phosphodiesterase ring and you degrade all the cyclic AMP, in which case then your concentration profile as you go down will look like this. Right? And you have localized areas, okay, of cyclic nucleotide concentrations in the cell. That was a make-believe thought experiment, okay? There's no slide. I don't know if that occurs, but now you think about the same thing, and these do occur, where I keep talking about the dendritic spine really behaving very differently than the dendrite that the spine is sitting on. It's going to respond to a different set of neurotransmitters. Okay, it's going to respond with a different level of intensity. Think of the axon hillock. Okay, you better not let all of the metabolic machinery of the axon hillock be wandering around the cell body and down the axon, because then you're going to have spontaneous action potentials firing all over the place. You don't want that. So you're going to restrict it. You're going to use your cytoskeletal system, but you're also going to have to limit the diffusion of your soluble second messengers. So you're going to take up calcium. You're going to degrade cyclic AMP. You're going to create a spatial profile of the second messenger. Okay? That's where this thing gets really sophisticated. And all of that is happening in this system. That's all that this says, basically. This is a nice picture from an olfactory bulb of different phosphodiesterases on different neurons. And as I point out, some of the neurons actually have more than one phosphodiesterase in it. But I just thought this is a beautiful picture. This is all regional, and this is within, these are all neighboring cells, but they're all using different phosphodiesterases. Okay? There you go. That's it. Good. 5 minutes early.